But yeah, thanks everyone for stopping by. My name is Rayo. I'm currently a bioinformatics programmer at UCSF. Title of my talk is going to be on enhancing reproducibility in immunogenetics, but it's going to be more about how I've personally leveraged containerization on bioinformatics workflows. So I work for the Hollenbach lab at UCSF. Our lab focuses on understanding the contribution of genetic region like HLA or MHC into different types of diseases, specifically like autoimmune or immune disease. But today's talk is not necessarily going to be about the science of behind immunogenetics or the tools that are associated and trying to understand these big questions, these big science questions. But I'm going to tackle more on the reproducibility issues that the scientific community is currently facing. And I also just wanted to preface the talk by acknowledging that there's actually not a lot of novelty on what I'm presenting today. I think my main hope for this talk is that I can raise the awareness for developers to think about how can we make our tools more accessible and reduce the barrier of entry when it comes to containerizing your workflow. I'm sure some of you are already familiar with the idea, and I guess I just wanted to break it down further so that it's not as foreign as we think it is. So I kind of want to start off with this survey that was done back in 2016, where Nature surveyed 1,500 scientists, asking them whether there is a reproducibility crisis. And to my surprise, over half of them, 52% of them, mentioned that yes, there is a significant and crisis in science. And this is when it comes to recreating an experiment, reproducing a data, or generating a figure, knowing what reagent to use, etc. And so there just goes to show that there is a big problem with reproducibility in science. And just to showcase that issue in computational biology specifically, the NLM, the National Library of Medicine, hosted a workshop back in 2020 where they invited 25 trainees in groups of five to recreate a figure from a published bioinformatics paper. And surprisingly, none of them were able to reproduce the image, even though they have been given the necessary input for that particular pipeline. And just to put this in a bigger context, there has been attempts to assess how well we are doing in keeping computational science reproducible. And so I'm not going to go into each of this, but these studies basically try to run pipeline that are publicly available to see what percentage of like how many of them are actually runnable from one end to the other. So I'm just going to showcase the one on the most right here. Out of out of all the pipelines that they tested on the Harvard Data First R script, only about a quarter of them is actually runnable from end to end. And so there can be many reasons why we're facing such a complex issue, right? It's such a big problem, and one of the main reasons is because it's actually really complex. Bioinformatics pipelines are complicated. You utilize multiple tools, typically tested on a very specific version, and it's usually dependent on what's the operating system of the developer. And so one way to potentially address this issue is by specifying the requirements. You've probably seen some of this on some of the GitHub repos that you've encountered in the past few years. You can specify, what tools you use, what version, how do you install them, etc. But it's not always simple. Sometimes when you try to install our packages, you need certain libraries installed, and those system packages or libraries are typically installed through your package manager, like yum or apt, and they usually want you to have root access or sudo access. And so that brings me to this idea of containerization, which I'm sure most of you are already familiar with, but I'm just going to give you a very brief overview. It is that containers packs every single tool, packages, and dependencies that you would like your users to have. So it will remove the complexity that comes along with your tools or your pipelines installation. And for the container platform that I choose, I actually decided to settle on Singularity. This was a tool developed by folks over at Lawrence Berkeley National Lab. which means it's meant for reproducibility in scientific computing. And one reason why I think Singularity is such a nice option is that it stores container in a single file, in a single zip file, that is easily shareable. Now, at this point, some of you might be thinking, why don't you just use Docker? It's been around longer. I think it's more popular. Um, it's probably used by a lot of software engineers all around the world. And the main reason why I picked Singularity over Docker is because Singularity is designed to be run as a non-privileged user. And this is especially helpful if you're working in an HPC cluster in a high-performance computing platform. For instance, in UCSF, we use what's called Winton, where it's like a shared resource. Everybody in the campus basically are allowed to access them. People over at UBC might be familiar with ARC, the Advanced Research Computing. People over at Berkeley, you might be familiar with Savio. UNSW, you might know Katana. So this HPCs, they typically don't let you have sudo or root access, which makes Singularity just a more appealing option compared to Docker. And just wanted to showcase one of the pipelines that we containerized in the past is what we call PING. It stands for Pushing Immunogenetics to the Next Generation. I have no idea who came up with the naming, but I think it's pretty creative. It is a tool that would let you do genotyping for the key region from short-read sequencing data, and it's just short read paired end sequencing data. And I'm not going to talk too much about the actual pipeline itself. What I wanted to highlight now is actually the three tools that are critical in this pipeline. And most of you have probably worked with one of these three tools before, or maybe all of them. And so now I kind of just wanted to walk us through the process of how do I create a container with these three tools? Just a very simple, basic container. And you can actually do it by instructing the container by letting them know what package you want installed and what version you want it to have in what's called the definition file. So typically, I just have the definition file sitting in my GitHub. It's similar to a Docker file for those of you who are familiar with the Docker system. And essentially, this is just like a text file. And if we go inside the definition file, it's gonna look something like this, where I set it into two different parts. So the first part is to set up the environment and the dependencies. Um, you can install tools that would help you ensure proper installation of other bioinformatics tools. So this would be things like wget, make, visit to, so extracting, getting all the tools, etc. You also would want to install libraries or system packages that are needed for bioinformatics tool installation. So in this particular case, libtbb def, this is needed for Bowtie. And so these are the kind of things that you want to take care of before you send off your tool or publish your tool to the users, so that potential investigators or users who are interested in your pipeline, they don't necessarily need to install all these different packages. We just trying to make their life easier. And the second part of the definition file is probably something that you're more familiar with. This is where you actually get the tool, extract them, and then make the tool itself. And these two parts that I broke it down into, it's just the more important chunk of the definition file. There's more, but I'm not going to go over everything. But in essence is that you can just specify your system packages, and then you can specify what actual bioinformatics tools do you want to have installed in your container itself. Once you already have your definition file, the next step is actually to build the container. So building the container means you want to get this CIF file. So this is the actual container file that I was mentioning earlier. Why I like Singularity is that it's just one singular file that tells you, hey, this is the exact container. And the command is pretty simple. You can just do like what's called the Singularity build. And let's say you have like your pipeline that's typically running locally, right? Like in this case, ping was written in R, And so it's going to be just our script and then the name of the script. And the way you want to use, if you want to run it through this container, you can actually use both the CIF and the input FASTQ as inputs in the command line. And if this is how it looks like when you're typically running your pipeline, it's just R script, or if it's like a shell script, then just sh or bash followed by the name of the script. All you need to do is have like 3 additional words in front of it that basically says, hey, I want you to execute my pipeline through the container. It's as simple as that. So one question that popped up as I was working on this is that why can't I just upload the .zip file to the repository? Like why can't I just upload this? Why do I have to go through building the container, or like why do I have to give instruction for user? Why can't I just put the zip file in my GitHub repo, for instance? And the main reason is just mostly size limitation. If you try to push sizes that are like over 100 megabytes to GitHub, it's going to complain. Some of you might be familiar with the Git LFS. It will let you push file up to 2 gigabytes, and that's still a limit. The size of containers that I've worked with, the zip file, it varies, but I've encountered containers up to like 4 and 5 gigabytes. So, but even if your container is less than that, I think it's generally recommended for you to not have your GitHub repo to be bloated in terms of storage. GitHub typically recommends you to have it less than 1 gigabyte in terms of total size. But if you still want to upload it, your .zip file or your container somewhere, Singularity has something similar to Docker Hub. It's called Scilabs, where you can push your image, and then whoever wants to use it, you can just pull it right away. The only downside is that it only has 11 gigabytes worth of storage. I think if you pay more, you probably get more storage. But yeah, this is just like one of the alternatives, instead of actually making the user build their container itself. So another question that pops up as I was developing, different tools and containerizing is that what if I need multiple Python in my pipeline? So in this case, there are 2 amazing tools, Amos and Ragtag, that require a different version of Python. So Amos needed to be installed in a Python 2.7 environment, whereas Ragtag is like in Python version that are like greater than 3. And this is actually a problem with one of the pipelines that we are developing in our lab, which is called Image Constructor. This was developed by Kristen Wade, one of our wonderful postdoctoral scholar. This is a short-read de novo assembler for the MHC region. So for those of you who might not be familiar, MHC is actually one of the most variable region in the human genome, which makes the problem of de novo assembly even more confusing. But in distal in particular, that's what we're trying to address. And this is where we actually use those 2 different tools that I was mentioning earlier, the one that has 2 different Python versions. And one way to address this is by using what's called utilizing something that most of you are probably familiar with called Conda, where you can differentiate your system Python with other Python installations. So you can have, Python 3.7, or you can have Python 3.8 installed in one of the environments. You can also do this with other tools as well that are offered by Conda. So you can have multiple R or Java version in different instances of environment. And so integrating Conda and Singularity is actually a viable option. And I think the concept is pretty basic in that inside your Singularity container, you're just going to have your first Conda environment where you want to specify when you're creating that environment, like, hey, I want it to have a Python 2.7. And so that being said, you will be able to install Amos, and you basically would just do the same thing for a second Conda environment where you specify the version of Python, and then the tool that you want installed. And the nice thing is that you can actually, there's seamless switching between one Conda environment and the other. because the pipeline that we design is meant to run from end to end. In the middle of the script, we can easily just switch from one Conda environment into the other, and so we can just use Amos and Rectech in places where we need it, basically. And so that brings me to my conclusion. The first one being that Singularity offers a reliable solution for bioinformatics software that is catered for use in HPCs. So HPCs are probably utilized by lots of researchers from all around the world. Utilization of container platform removes the need for investigators to install multiple dependencies. So I personally, as someone with a little bit of bioinformatics background, have struggled or been frustrated trying to install like a really interesting tool. But I can only imagine what would be the case if someone who is not necessarily inclined in terms of software look at our tool and then are interested in it, but then just struggle trying to install different libraries or system packages. So by containerizing, we kind of like help them along the way, try to make things as simple as possible, And my third point is that a more complex pipeline that requires multiple versions of the same package can be containerized with the help of Conda. In the example, I showed you multiple Python versions, but you can actually do it with other tools that are offered by Conda as well. And just a reminder that doing all of this, it's basically to ensure that the integrity of our research are going to be preserved as time progresses. That being said, I just wanted to acknowledge people from my lab, for Kristen and Wes specifically, for the amazing pipeline that they developed. I have here the QR code for these pipelines if any of you are interested in either a Kier genotyper or an MHC short read de novo assembler. And I also just wanted to acknowledge that this is not necessarily the best or the most optimum way to deal with reproducibility issues. So I would also love to get some feedbacks and or suggestions on how to turn this into a better effort, because that's really what I'm trying to advocate for. And if you would like to containerize your workflow, but you felt like it's overwhelming, I have my email listed here. Feel free to chat, and I'd be happy to help you throughout the process. Thank you.
Thank you so much, Rayo. With that, we have time for one question for you. Does anybody have questions? Someone is walking to the mic. Hang on a second. I don't think it's on. Thank you.
Great talk. I was just wondering, so it seems like you're a proponent of using one container for your whole workflow. Why do you see the advantages and disadvantages versus using multiple containers, one for each step, and then constructing a workflow, for example, in WDL or CWL to define exactly your whole pipeline?
Yeah, this is a very good point. I think the main reason why I like to keep things in one container is just for the sake of simplicity. So I know what people without like a bioinformatics or like a software background typically struggle when they're trying to utilize different tools. I like to have just one Singularity container because the instruction for you to just build and run the pipeline is fairly simple, but I have not yet encountered any technical challenges that would require me to use multiple container. If in the future there's pipeline that I would need to containerize in multiple containers and then use like a CWL to integrate them, I think that would be a great idea as well. And it would be a great effort to see how well can we document them so that it's easiest for people to access our pipeline.
Thank you very much, Rayo. Another round of applause, please. Thank you.
Thank you.